everyone, my name is Nahda Ahmad Rashid, actually master student in the Department of Fundamental and Industrial Microbiology in Istanbul University. I'm here to share with you a part of my result of my master thesis entitled as Caving Impact on Bacterial Diversity, a case study of camping in Morja Cave. Studying cave microbial diversity is a challenge due to the physicochemical properties of caves, which are strictly different from those of the Earth's surface. With the culture-independent investigation method, the cave microbiology has known a new era and further microorganisms have been identified. It is reported that microorganisms play an important role in some cave formation as well as cave structure formation. It is reported that microorganisms play their role in the solution and precipitation process, which result in the deposition of speleozymes like moon milk. Adaptation of these microorganisms in such extreme conditions such as darkness, low but stable temperature, limit of nutrient and others constitute the most attractive character of microbiologists in this environment. This adaptation reveals metabolic reactions which used enzymes and other bioactive compounds with industrial and biotechnology potential use. For the cave ecosystem activities and for a permanent bioactive compound source, protection and conservation of the cave microbial diversity is unconditionally important. How human can impact on cave microbial diversity? It can be through touching cave walls and structures, bring excite microorganisms, using lamp for illuminate for the cave as the cave is a dark ecosystem, introducing of organic materials into caves through food and other, fire for warming can augment or increase carbon dioxide and temperature, improvement of cave infrastructure like construction of paths, cut off cave speleozymes, manipulation of cave pools or rivers can impact on cave microbial diversity. My study site is constituted of a campsite installed inside Morja Cave. What is Morja Cave? Morja Cave is located at Middle Taurus Mountains with the other two deepest caves in Turkey. These mountains constitute the Tasheli Plateau, which is about 900 square kilometers. In 2019, this cave is estimated theoretically to be reached to 1,600 meters depth. Sediment and surface samples were taken from different points of the cave activities during the cave expedition in September 2019. The impact of caving in cave microorganisms is studied through samples taken from various points around the camp, installing during 4 days at 1,040 meter depth of the cave. Every selected point has been investigated through 2 times, before the camp and after camp. So I had 12 samples taken from 6 areas. Surface sample and sediment samples taken from the campsite, different sediment samples taken from toilets area 1 and 2, and surface and sediment samples taken from points far from the cave activities area. Once they reached our laboratory, the DNA isolation from the samples was done with the fast DNA spin kit for soils following the manufacturer protocol. The isolated concentration were checked in nanodrop apparat and controlled by run the PCR product in a gel electrophoresis. After that, the V4 and V5 variable region of 16S rRNA genes from the purified isolated DNA were amplified with the addition of bacterial specific universal primer. After that, sequencing and bioinformatic analysis comes. Amplification control is done through gel agarose run of the PCR product, and purification of this amplicon was done by using the impure XP kit, especially using the bead found in this kit. The purified amplicons are prepared for sequencing according to the protocol of the used kit. Service procurement was carried out with an average of 50,000 reading for each sample on the MISEC Davis for next generation sequencing service. The bioinformatic analysis, the quality control of all obtained sequence is through the FastQC program, and 16S rRNA amplicons data are analyzed using QMES software. The alpha diversity analysis of the samples was determined using the O2 table with the QMES software and the Rawan, Shannon, Simpson, and observed O2 as index, indicating the diversity levels were calculated. The result, the alpha diversity result observation, the observed O2 species is between 219 to 979. The most O2 species is observed in the NAR14 of the sampling area F, and the lowest O2 species is observed in the NAR12 of area E. Also, the highest bacterial richness and diversity were observed in NAR14. Considering all our samples, the richness and diversity increased during the CAM period, except in the area A, where the diversity has decreased, and in area D, where the richness has decreased after CAM period. For the taxonomic result, in the phylum level, we constate that Proteobacteria is the most dominant phylum in all the samples taken before CAM, and the NAR4 and NAR10 which taken after the CAM. But Firmicutes dominate all the samples taken after CAM except in NAR4, the area B after CAM, and NAR10, which is the toilet area 2 after CAM. In the genus level, every sample of every sampling area showed different rate of identified bacteria. Some bacteria identified or dominated in samples taken before CAM disappear in samples taken from the samples after CAM. Some of them appear with high rate, others show a decrease in the rate. Here we can retain 3 points through our result. First, the specificity of every area in CAM is more evident here, since every sample before CAM showed different bacterial diversity or different bacterial structure. 2. A decrease of the diversity is reported to be a cause of impact on microbial diversity caused by human, By this way, we can resume that only the area A, which is the surface area of the campsite, has been more impacted by the cavers, even if no human indicator bacteria has been observed in this study. Increasing of Acinetobacter number in the surface area of the campsite can be caused by the introduction of new Acinetobacter diverse species related to human microbiota, which is the species Acinetobacter leofi, which is a human normal skin microbiota. I will conclude my presentation with perspective of cave microbial protection. We can protect the cave microbial diversity through limit of cave visitor number and hours. Cave managers should avoid artificial improvements inside the cave. Education about the importance of caves and cave microbial diversity should be exposed to people through conferences or other education systems. Collaboration between cave managers and cavers and scientists, especially the microbiologists, is very important. Education of cavers as well as visitors is very important. Also, cave should have a rest period. Every cave should be used as specific fortune and avoid bad habit. Pool, rivers, and other sources of water should not be accessible for cave visitors despite for sampling or for scientific studies. Camp should be in a small part and during a limited period. Cover clothes and water should be stand clean. We should be careful that every touch or work step is acceptable to disturb the invisible microbial cave structure. Use of fire for warm-up or using cigarette inside the cave should be banned since they increase the carbon dioxide, organic matter, and toxic matter inside cave. Finally, encouraging the microbiology studies of caves. Also, without human investigation, the positive or negative impact of the nature remain in the dark. The conservation of this natural source should be studied before any activity. For the life of caves as well as the continuity of industrial and biotechnological natural resources, we should elaborate cave using protocol. Thank you for your attention.